It's so great to see all of you. I really want to thank you for coming. My name is Matt Vanderheiden. I'm the director here at the Koch Institute for Integrative Cancer Research at MIT. And tonight, we have a very special With Insight program. We're going to celebrate a special milestone. The approval, as those of you who are here and just saw, of a new treatment for bladder cancer. And this is approval for a therapy that would not exist if it not had been for research here at the Koch Institute. Now, as a scientist, as a researcher, I have to say one of the most rewarding things that can ever possibly happen is to think, wow, something I did actually made a difference for a real patient, really impacted a person's life. And the video you just watched, it's exactly like, look, this is a guy who now life has changed because of work that was done here in this building. And tonight we're going to hear a bit about what led to that, how this new treatment, INLEXZO, came to pass, came from an idea that existed in Michael Cima's lab, ultimately to now treating real patients. Now, MIT in general has an explicit commitment to not just doing amazing science. It's important to me. I think we do amazing science here. But it's not just doing the amazing science, it's actually translating it out and making a difference for real people. So the whole MIT motto, for those of you who are alumni, you know the Men's motto, right? So, mind and hand. I'm very proud that us here at the Koch Institute, MIT's cancer research community, really embraces this. I like to remind our scientists all the time that the work we do, yes, we can easily get caught up in the academic stuff that we all deal with in our daily lives, but patients are counting on us. Patients are counting on us because the discoveries we make a real difference, make a difference for people with cancer. And on the times when we get a success, like INLEXZO, we want to celebrate those successes. Now, we've made many discoveries here at the Koch Institute. I'm really proud of that, many of which have contributed to real impact for patients. Of course, we've been around now for more than 50 years, so some of you have been here. We've celebrated these milestones in the past. Over that time, my predecessors mapped many of the genes that are important for causing cancer that contributed to the development of drugs that target some of those mutations. But then about 15 years ago, my predecessors had this visionary idea to found what is now the Koch Institute, this new multidisciplinary model of collaborative research that really brings the best of MIT engineering together with traditional cancer research. And for those of you who are joining us for the first time, that's really what's special about this place. This Koch Institute, the building you're in today, is really a mix of biologists and chemists, materials and scientists, chemical engineers, biological engineers, data and computational people. And this mix of expertise has brought exactly what we're here to celebrate tonight. That is, this now brings the best of MIT engineering and technology development to bear on some of the hardest problems in cancer research and leading to new solutions that help patients. Now, there's lots of stuff we do here at the Koch Institute, and if you're a regular attendee of our With Insight program, you get to hear about these things. AI imaging for early detection, improved drug delivery, ways to target cancer that we didn't think were possible before, new ways to predict which treatments are going to work. However, each of these solutions I mentioned is really the culmination of hard work by real people working in laboratories upstairs. And what's special about tonight is that we can sort of get an inside look into how the curiosity of the people upstairs asking questions, what tools and mechanisms might help diagnostics be better? How can we improve immune responses to destroy cancer? Can we come up with new machine learning or AI tools to accelerate research and help patients? Or can we, sorry, I did that by mistake, can we come up with a new device that delivers drugs in a way that now allows real patients to have much less morbid treatment and basically continue living a much more normal life than they would have had to prior to the invention of some of our therapies. Now, we're going to hear about this solution tonight. First, we're going to hear from our presenters. We'll hear from Michael Cima, who's going to tell us really about where this came from. And then ultimately, we're going to have a panel discussion. I have a couple questions I'm going to ask them, sort of set the stage for what's really happened. But I want also you guys to think about which questions do you want to ask, because we'll give you guys some time to really ask these guys, tell us more about INLEXZO, where it came from, why they're excited about it. And so without further ado, let me invite my colleague, Dr. Michael Cima, up here to the stage. Dr. Cima is a professor in MIT's Department of Material Science and Engineering. He's also a David H. Koch Professor of Engineering. Since 2009, he's been faculty director of the Lemelson MIT program, which is a program, for those who don't know, who tries to inspire youth to be inventive nationwide. He was appointed Associate Dean of Innovation for the School of Engineering a few years ago in 2018, and he served as co-director of MIT's Innovation Initiative. Michael is an elected member, a lot of important things to say about you, of the National Academy of Engineering, also the National Academy of Inventors. He's a serial entrepreneur, so he's co-founded several companies, most recently Strategen Bio, which is developing oxygen sensors for customized cancer therapy, also a very cool story, maybe a future with insight. But tonight, Michael has really achieved, I think, what the pinnacle is of really any of us in science. He's made a discovery that has ultimately been approved and now makes a difference for real patients. And Michael, welcome. Congratulations.
So we just have a few slides. I'm going to introduce Karen Daniel here in a moment, who will get up and speak briefly about some of what we've worked on together for many years. That was a great introduction, Matt. It made me think of something I get asked all the time by students coming to MIT. They have a good sense of what great science is before they come here. You know, it's at the boundary between what you know and what you don't know. But they always ask, what's great engineering? And I have to tell them it's similar in that it's at the boundary of what's possible and what's impossible. And given how many times I was told this project would not work, I think maybe we're got an example of great engineering here. So I will tell you a little bit about it. So we're going to introduce, as the video had, a little bit about, maybe I have to stand over here because I can't see what I'm pointing to. Bladder cancer. In particular, we're going to talk about non-muscle invasive bladder cancer. And we're talking about as much as 85,000 new cases a year. It's primarily associated with smoking. So about half these patients, you can say they had a history of smoking. Not a lot has changed in the past 30 years in treatment of this. What's primarily done is a BCG. You heard about that in the video. BCG is tuberculosis bacteria. And what they do is they instill a dispersion of the bacteria into the bladder of these patients and basically give them a urinary tract infection. And the idea is the inflammation that's generated attacks the cancer. It was one of the first immunotherapies, if you can call it. Not very specific, obviously, but that's what it is. And the problem is that there's a lot of recurrence in these patients. In fact, when you say that 85,000 new cases a year, in actuality, the number of people being treated, it goes, because they keeps on recurring. So they're treated for decades. They're under surveillance. It's closer to something like 700,000 patients in the United States at this time. So it's almost tenfold higher are being, as far as prevalence goes, how many people are being treated. BCG has another problem. Not only is it not effective in all patients, but it's actually hard to produce now. In fact, since 2012, it's been a shortage and it's hard for people to get access to it. Then the other problem, of course, is that those people that don't respond are categorized as very high risk. And their options really are only to have their bladder removed. And if they're not healthy enough to have their bladder removed, then these people ultimately succumb to their disease. So now let's go back in time. My research involves, in many cases, what I call single compartment delivery. By 2005, I'd worked with many pharmaceutical companies in the development of molecules that ultimately you'd hope become drugs. The problem with that, as I realized, is that you make a lot of compromises in the design of those molecules because it has to get to the target, let's say the tumor. It has to not go other places. It has to be eliminated quickly. It has to do all of these things that are really compromises in the design. And so I had the idea, working with many different projects, to design products that put the drug where it needs to be. So typically that would be in the compartment of the tumor, in the brain, in the muscle, in the peritoneum, in the eye. Have many examples like this that I've worked on. And bladder turned out to be a really good one because urologists are already making drug solutions and instilling them into the bladder. There's already a procedure associated with it. And I studied those procedures, and with Karen's help at the time, when she was in graduate school and several others that I'll mention here in a second, we studied those procedures and how physicians actually did this. And we came up with the idea that what we would do is put a device in the bladder, and this is a very early one, that actually didn't work. This would get folded into a catheter, and it's drug devices. In this case, it shows pieces of lidocaine in it, drug pellets inside of it. There's a laser drilled hole in it to allow the elution of the drug. Medical grade silicone has the property that it permeates water. So when we put this into the animal, there were rabbits at the time, the urine in the bladder would permeate through the medical grade silicone, dissolve the drug, and then make an osmotic pump and push it out. The problem was that when you fold this up into a catheter and push it out into the bladder, it could immediately come out of the bladder because the detrusor muscle of the bladder would squeeze it and it would go back to the shape that it had in the catheter and just be eliminated. We saw that in the very first rabbit that this went into. Ultimately, what we did with Hee-Jin Lee, who was a graduate student at the time, he was a mechanical engineering student. We designed a spring that looks like this pretzel shape. Ultimately, the product looks like this. The spring has got a super elastic alloy in it that's heat treated to be this shape that retains it. And then a drug reservoir is carried on it. In the real device, it's actually a dual lumen silicone tube that's in the commercial product. One has the nitinol wire in it, and the other has the drug in it. But it works the same way. And so this is what the INLEXZO looks like. It's placed into the bladder. You'll see a little video clip here through a catheter and pushed out. And it stays in the bladder for a specified period of time. The urine does exactly what I described. It permeates through the medical grade silicone and then dissolves the drug to produce an osmotic pump inside osmotic pressure, which forces the drug out through the small laser drill hole. You can put this device in a beaker and measure how fast the drug comes out. And you do this same thing in a person. And the remarkable thing, it's identical. The in vitro in vivo correlation is exactly that. And amongst the different variables that you can use to adjust the release rate, the most important one is probably the surface area of the device, because you design it to be the rate limiting step for pushing the drug out. Now, I always get asked, how do you get it out of the bladder? And that turns out to be this device here, which is a cystoscope. It's the second most common urological procedure. It can be done, both are can be done in the doctor's office. These take seconds to do. And what it is looks a lot like a catheter, except it has an optical lumen that can light the inside, and a working lumen that you can put a tool through that has a little gripper there, and you can grab the device and pull it out. That's what the urologist does. They're called forceps. But this is a deployment. Catheter looks like that. Push it out. This is why we need the super elastic wire. And then to go in, with the forceps, you go in and grab one end and pull it out. I think deployment is on average 60 seconds and removal is 90 seconds or something like that. Yeah. And it's done in the exam room at the doctor's office. Doesn't require an operating room. I'm going to now introduce Karen. Now, Karen Daniel was a graduate student at the time. She was actually co-advised by Bob Langer, right? But I worked with her very closely during her graduates. Her project was a diagnostic project. She was doing an MR detector that I won't talk about, but Karen was a chemical engineering graduate student. That's the connection to Bob, of course. But there were several other graduate students at the time that I want to mention. Hee-Jin Lee, which I mentioned before, PhD student in mechanical engineering. The Hong Lin Hoduk, who is a material science PhD, and then Grace Kim, who was an HST student. That was a significant fraction of my group at the time. So you can see it was a rather eclectic group of students. And Karen the four of them went to the company when we started it. And I didn't mention that. I should have said something about the timing. Oh, it won't go backwards. So the paper the first project was started in 2005. That was funded by philanthropic sources. It wouldn't have been funded by the NIH. It just would not. It tried. And and by 2008, we started the company based on that data. And the company was called Taurus. The four students that I mentioned went to the company after graduating. And two of them still are there. And Karen has grown to be a recognized leader within J&J. I can speak because I've talked to many other people at high levels in the company, and they all speak very highly of Karen. So one of my proudest achievements are people like Karen Daniels. So, okay, there you go.
Thank you. I always tell people that working at Taurus in those early years was like a continuation of grad school, but just with one project and a lot of people helping you get that done. But we would be working on things, and every once in a while Michael would stop by and he'd look at our data and he'd say, "Oh, have you thought about trying this?" So it just felt very much like grad school. And also I was telling Hee-Jin the other day that when a new person joins a company and wants to learn about the system, I still say, you really should read Hee-Jin's thesis. I'm just pushing that on people all the time, because that was the foundation of what Taurus was built on. I'm just going to spend a few minutes talking about bladder cancer and what we are treating. So this schematic here is just like a cross-section of the bladder wall. And as you move from the left to the right, you can see you go from non-muscle invasive bladder cancer, where really the tumor is just in that urothelium that touches the bladder urine. And then as you move to more muscle invasive, it goes deeper and deeper in the bladder wall. And then by metastatic, it's throughout the whole bladder wall and also in other parts of the body. And often what happens in trying to treat bladder cancer is a company finds a drug that they can deliver systemically, and it works well on metastatic bladder cancer. And so then they say, well, then let's just give it to patients who are earlier in the progression. And that hasn't worked well because in order to get concentrations through the bladder wall, from systemic through the bladder wall to the urothelium at efficacious concentrations, you have to get really high doses systemically, and you don't have good tolerability with that. And so, as Michael was saying, another approach is to do instillations where, okay, we're going to put the drug in the urine, give the patient a solution of that, and then they just have to hold it in their bladder for as long as they can, maybe an hour, maybe 2 hours, and then that's their treatment. But because that's not really enough duration to have a good impact, they have to come back every week to the physician's office. So, it's hard. Sometimes these patients don't live close to the physician's office. So like the travel back and forth, it's not a good impact on the patient. And then also they have such short dwell times for that drug that you don't have good efficacy. So that was the idea behind this pretzel, is you put it in the bladder and then it can slowly release the drug over time. And so we, as Michael was saying, we developed this system for delivering gemcitabine. And now gemcitabine was not the first, it wasn't the only drug that we considered. We were looking at several different drugs. Taurus was not in the business of making new drugs. We were taking existing drugs and changing the route of administration. So we were looking at several different drugs for treating bladder cancer. And we were looking for something that, if you put it in local, it's going to be well tolerated locally. You don't want to put something in and have that compound like make ulcers of the bladder and cause difficulty there. You want it to stay local. You don't want it to go into the bladder and immediately go to systemic circulation, because there's no point of treating locally if you're going to have systemic exposure. And you want to have a good partition from the urine into the tissue. Like if you put the drug into the urine and it just stays in the urine, you're not doing anything. So that's kind of the criteria we use to narrow down to gemcitabine. And as Michael was saying, it's a very soft, flexible, we can come touch this after if you want to. The ends are very soft. We really cared a lot about patient tolerability. There had been other attempts at local delivery to the bladder and it wasn't well tolerated. The patient wouldn't keep it in their bladder long enough. And so that was really what we were focused on. And something I want to point out is that we have in this pretzel, the gemcitabine system, we have 225 milligrams of gemcitabine, and that's released over about 7 to 10 days. And the installation of gemcitabine that was sometimes done uses 2,000 milligrams of gemcitabine for just like 1 or 2 hours. So we are able to really drastically reduce the amount of drug needed and just extend the exposure to get that efficacious concentration in the bladder wall. And here on the left side, it's just a little bit more about the osmotic system. What we found was, well, actually, we start at the graph on the right. That blue bar on the bottom, a urine concentration of 4 to 5 micrograms per mil, that's what we wanted to stay above. That was what we had as our minimum effective concentration. So we wanted to be above that. And when we made our pretzel and we just filled it with gemcitabine, we were not releasing fast enough to keep above that level. So what we found we had to do was we had to add some extra tablets into the drug core that would act as like an osmotic engine support. And so we ended up using just urea tablets. Urea is already present in the urine. It has a really high osmotic driving force. And then the blue squares shows the concentration in the urine of gemcitabine with this pretzel. So we were able to stay above that minimum target concentration for up to about 7 days. And when this system is used to treat patients, it remains in the bladder for about 3 weeks because gemcitabine will not only target cancer cells. It would, if you have exposure to healthy cells, it will also damage healthy cells. So it just releases for 7 to 10 days, And that has an impact on the high turnover cancer cells, but it doesn't have an impact on the healthy cells. So then the patient has a 2-week rest period where the system is still in their bladder, it's well tolerated, but it's not releasing any drug at that point. And so then at week 3, the patient goes back to the office, the system is removed and a new one is placed. And then on the graph, that green spike, that shows the concentration for the installation. So the peak concentration for our GEMRIS system, our gemcitabine system, is about 40 micrograms per mil. And then for an installation, it's about 40 mgs per mil. But what happens is a lot of that gemcitabine in the urine ends up being wasted because it cannot be taken up into the tissues. And it's not there for a long enough time to have the impact that it needs. And another thing about the bladder that makes it well suited for local delivery is that in healthy tissues, there's very tight junctions between the cells. And the bladder is really designed to get rid of a lot of things. So there's a lot of compounds in the urine that are not going to go back into the tissue in healthy cells. But there's a research paper that showed that, and a lot of papers have shown that when there's cancer present, cancer makes those tight junctions looser. And so then it increases the permeability of those cells. And so, if you remember back to the schematic of the early non-muscle invasive bladder cancer, what's just in the first 2 layers maybe of the bladder wall, so you will be able to treat those cancer cells without it going all the way through the healthy layers underneath it and going to systemic. So in that way, the bladder is really set up well for local treatment in oncology. And then the plot on the right is just an example of, just a schematic of some potential different release rate profiles. So, we want to stay above the minimum effective dose, below the maximum tolerated dose, and that first order release kind of looks like the profile of our gemcitabine system. But, there might be other indications or other drugs where we want a different profile. And so Michael was talking about the parameters you can change, and, we can tweak different things, and we can have, a more zero order profile if you wanted to. And that leads me to my last slide, where again, this is very much like the schematic of the progression of the bladder cancer, non-muscle invasive on the left, and as you move across to muscle invasive and metastatic. And you can see here the INLEXZO logo. INLEXZO is the first approved commercial product for the gemcitabine system, and it treats one slice of the population. But by taking the system and using it as a platform and changing the drug that goes inside, we're able to then target patients in different parts of this bladder cancer journey. And those are depicted by the Moonrise study. So that's a system that uses a more targeted API, and it's able to be in the bladder for 3 months at a time because it's only going to target the cancer cells. And so we have, different systems to try to target all the different phases of the disease. And as we, continue these clinical trials, we're learning more and more about the disease. And the INLEXZO Sunrise 1 clinical study is the data that was used for the FDA approval last year. And that showed a complete response rate of 82%. And when Taurus first started about treating bladder cancer, we were saying wow, it would be great if we could get a complete response of 60%. That would be amazing. And then, we find that with our first clinical trial population, a complete response of 82%. And so we really think that this will be a practice-changing system for these patients. And the last thing I want to talk about is just kind of the timeline. So Michael is saying that Taurus was founded in 2008. The first time we started working with gemcitabine was in 2011. By 2015, we were ready. We had all the data we needed to start going into people. We were acquired by J&J in 2019. And at that point, we were wrapping up our phase 1 clinical trials where looking at safety and tolerability. And then we finally got approval for the first indication in 2025. So that's 14 years, of going. And at the time that I started, I thought, you just need a good product. But I've learned that along those 14 years, there's so many ways it could have failed. You need innovative people along the whole production side of it. So I think now we'll move to the question and answer part of the talk.
Thank you, guys. Let's move on over here and
Karen, will you
we'll
send them
have a bit of a discussion. So for those of you who, technical check here. So Michael, Karen, thank you so much. It's really, truly inspiring what you guys were able to accomplish. So just for the audience, think of your questions, because we'll get to those here in a minute, but there's a few things that at least I'd like to cover. And so Michael, I'm going to start with you. And I guess as an oncologist, I can appreciate what the problem here was, and I will just say the 82% complete response rate, like that's incredible. You don't see that very often, right? That's a big deal. But you're a material scientist, right? What led you to this in the first place? Why this problem?
Well, as I said, I was working on single compartment, drug delivery. That was the hypothesis, or the method of change. And I got introduced to a urologist. And I found out that urologists had this crazy practice of instilling drugs into people's bladder for all kinds of diseases. They do it off-label and stuff like that. And it just seemed that was perfect. And actually, the first thing that came up was bladder cancer. And as you can see with Karen's last slide there, it's a pretty complicated disease. So we didn't start there at the company, but we knew we had to go there. We just had to figure out a path. And we got the right people together and really had it was just a fantastic group of people that could attack a problem like that and do great science and great engineering at the same time.
Yeah. Well, Karen, let me ask you, because you were there, right? You were a grad student at the time, right? You joined this guy's lab, and what was the pitch? Someone's saying, we're going to instill stuff in the bladder. How did that come to pass?
I remember going to talk with a urologist at Boston Children's, and he was
at
saying,
Children's, so these are kids.
Yeah.
Yeah, well, but he was saying there's just we want to be able to do better treatments. He's like, but this is all we have to work with, And he was saying it's just he was saying we are so limited by what we can give our patients, and it was really his passion for finding better treatments. As someone who was in the field and knew what was available, and saying we have to do something better.
And who came up with the pretzel shape, right? I mean, that makes a great story now. We're putting pretzel-shaped stuff in the bladder.
Yeah, it was
that sounds good.
Heejin and I.
Where did that come from?
It came from a mistake. I mean, like I mentioned that first, the reason why I put that early device up was that it went into the rabbit. I mean, the very first time Heejin put this into the bladder of a rabbit, it came out within seconds. And he literally walked from the animal.
And it was it was
that big loop.
A big loop.
It's not the pretzel shape. And it just filled it up into the catheter, pushed it out into the bladder, and of course it comes out like this. And then the rabbit just decided to void and pushed it right out. And Heejin walked from there to my office and said, "We're toast. It's not gonna work." And it was totally despondent. And I said, "No, Heejin, if we can solve this problem, we have an invention, because this is the obvious
it's not
thing."
Obvious. It's
yeah,
not
the
obvious.
obvious thing didn't work, and that's where we worked on designed a spring that would allow us. So the big issue was we wanted it to be soft, but if the detrusor muscle squeezes too hard, the two ends of the two sides of it go like that, and now the spring constant went up by a factor of 4. And so now it's hard. So if it's if it's not being compressed, it's very soft, moves around the bladder. That was the other thing.
Yeah.
And it had to not stay in one location. And this stayed in. And it's stayed in people too. So it works. I mean, that's how that started. The mistake turned into an invention.
Wow. Wow. I mean, it's one of the many stories of good engineering. For the non-physicians in the audience, you think, wow, that hole's really small. It's actually incredible how big it is. I mean, catheters going, like there's actually more space there than you would than you would think. Although I have to ask the question, because I have to say, you addressed it, but the removing it part?
Oh.
So how does that work? Because if it's the spring, like the spring piece for
The
removing it is the same thing. Do you actually have to pull it out in an uncoiled way? Is that how that works?
Yeah. Typically, well, urologists will do what they do.
Yeah.
You know,
I know.
But
I work with them all
the practice
the time.
that
Yes.
I was familiar with, they would grab one end like this and then pull it out like that.
Okay.
And but I'm sure they've grabbed it in
I'm
the
sure
middle.
it just
and
gets
pulled it out.
yanked out a lot too. Like,
You know, they're
they're the
physicians.
cowboys of the surgical world.
Exactly.
I can say that.
We did a lot of tests with instroms and saying like, oh, if you grasp it here and pull out through our fake urethra, grasp it here, you know, so that was in Heejin's
Yeah.
thesis as well.
I'm sure they broke those rules pretty much the first time they took it out. Yeah.
But you know, the important thing was by working with them right from the beginning, we designed it around the possibility that they wouldn't even have to, they could look at it and they knew what to do.
Yeah.
And that worked out to be the case. I mean, we asked them to read the instructions, but, but they don't have
Yeah,
to.
no one reads the instructions. Yeah.
Exactly.
You think like, because you don't read the instructions when you buy like a new electronic device, you plug it in and just fiddle with it to try to make it work. You would think your doctor would do something different.
Yeah.
I hate to tell you, they probably don't.
There was one time where in a small company, you kind of have to, everyone has to do all the jobs. And so one time I was going to train a physician at a new site to participate in our trial. And we would always send someone there. Like we'd bring our little placebo system and a catheter. And he was so offended. He's like, I went to medical school and now you're gonna teach me how to catheter? I was like, oh, no. Like, we assume you know that part. We're just gonna show you how to use this with a catheter. You know, but it was so intuitive and you know, that he was like, I don't need to be trained for this, you know. So.
And like I said, you know, those cystoscopies, to give you an idea how common it is, it's 3 million a year are done in the United States. 3 million, mostly diagnostic procedures, just to image the surface of the bladder, or do a biopsy or something like that. But so it's very common.
Yeah, no, it's very, very common. I suspect, I don't want a show of hands or anything like that, but I suspect there have been people in the audience who have had cystoscopies, and I'm pretty sure none of them would actually sign up to do it every day. So if this is 2 cystoscopies is better than more than 2.
I won't mention the venture firm, but doing the early pitches, people, we used to go to, and their partners would be there, and we would pitch this idea, and they said, well, what about removal? And I'd say, well, cystoscopy. And they said they got very uncomfortable and said, except for one, the partners said, it's not a big deal.
That's great. Well, there's a few surprises that we talked about there, but I don't know, I'm going to ask the question slightly differently in terms of, and I guess either of you should, or both of you should maybe answer this question. Like, I mean, my reflection on what you just said is that, look, great engineering, solve the problem, lots of sort of interesting and surprising things along the way. But for you and your journey of sort of seeing this over the last 20 plus years of, idea to now approved therapy, what surprises have you found? Like when you look back on it, what was the biggest surprise, the biggest challenge, the hardest part, like fishing for some stories here?
Well, I think for me, the biggest surprise is that every time, Taurus is a great example of that, but every time I've put drug into a compartment, I discover new pharmacology. And that's very true here too, because what's different about using gemcitabine in this way is that the patient remains immunocompetent. Most of the time, systemic delivery of cytotoxic agents, that you can't say that. And what we think actually is a contributing factor here is that these patients, because you're treating over a period of time, killing the cancer over a period of time, it's producing antigen, and those patients develop an immune response to their own cancer.
So this is also an immunotherapy like the BCG.
They call it immunomodulatory, but now that it's approved, I already know of people that want to do that study and prove that this is happening. And I've talked to a number of urologists that think that might be a great experiment to do. But we know it is, because like at the company, when we first had these observations, they came back and recapitulated this effect in an animal model and showed that, for example, you can ablate the tumor on the surface of the bladder by delivering gemcitabine to the bladder, and then get rid of that, and then go back a month later and try and grow that tumor in that animal, and you can't.
So I'm going to now put my genitourinary oncologist hat on and say, so metastatic bladder cancer is treated. It's one of the more successful indications for drugs that attack, basically modulate the immune system, so-called checkpoint blockade therapies. Is that a trial that's being done? I should ask you that, Karen. Is that a trial you guys are doing?
Well,
INLEXZO in the bladder and someone with metastatic cancer and treat them with immunotherapy?
So we are looking at combinations of INLEXZO with other things. Yes.
Wow, for metastatic disease.
I'd have to go
muscle
back.
invasive.
Yeah.
And mostly the muscle invasive. If you look at the draining lymph nodes, you find a lot of positive lymph. In fact, our first experiments were in humans were in patients with muscle invasive disease, and they were all going to be scheduled to have their bladder removed. And they looked at the draining lymph nodes of those patients, and there were much fewer than what they normally see. That was our first indication that much fewer positive lymph nodes than you would normally see. And that was the first indication that something may be going on, which was what prompted the experiment that I described earlier in the animal. And so I guess the biggest surprise for me is that you've got to keep on doing science all the way through this, because you're going to find things like that, which are going to lead to the next, it's a platform, so it's going to be into the next therapy and that sort of thing, and new indication. That kind of.
Yeah, no, Irina really highlights like just because you get something to where you think it needs to go, there's new things to learn and new possibilities out there. And I think sometimes people may not realize that that's the case. So Karen, I'm going to push you on the same question that Michael answered, and that is, like, what was the biggest surprise or challenge or any good stories you can tell about the journey here?
Yeah, I think I kind of alluded to this at the end of my talk, but I always thought of like the end, like, is the commercial approval. And it was like, yeah, that's what we're working towards. Like, that's okay, you succeeded. But then I went to like the commercial launch part of, like, when J&J was getting ready for that. And it's like, oh no, like, commercial approval is when all of their jobs start. Because if it's approved and no one uses it, then it's useless. So you have to have all these people to train and to like all the logistics of getting to all these places. So it was like, for us, we were thinking of it like that's the end of our job, but it's just someone else's job is just starting. And so I hadn't, like, you kind of know what you do and you know what you know, but there's so much
you don't
other
realize
stuff
there's
has
this
to be
whole
done.
arm of the industry that's job it is to,
and
this exists, otherwise.
I got to give a plug for J&J there. They've been a fantastic partner, because it takes a company like that to do all of that, to make sure patients can actually get this. There's a J code for this. I mean, all putting all that into place, you can't do that as a small company, and they've been just fantastic partner.
And because it's a drug device combination product, it doesn't fit neatly in anything that has been done before. Like there's lots of drugs and there's lots of guidances for that and there's lots of guidances for device, but that drug device combination section, like really involve, like everyone who touches this has to be innovative in their job because they have to figure out a new way to do things.
Was that a challenge in sort of the approval process, needing to
It was,
navigate?
yeah.
So for people out there who don't live in this world, there's separate regulatory processes for devices and drugs.
Yeah.
But this is a little bit of both, and so I guess you had to
it
do a
was,
little bit of FDA education.
yeah, it's regulated as a drug because that's what makes it work. But they then have like the device side as like input and would be reviewing it as well. And even within J&J, there's lots of procedures for developing a drug and procedures for developing a device, but it was like work to get those together early on.
Yeah, I think on the regulatory side, I think in this particular case, and it's a little different, but I will say that what they call chemical manufacturing controls part of the regulatory process, they like to apply the same thing each time, and they can't do that here.
Yeah.
It's not like making a tablet. And so there's a lot of discovery, even on their side, as like, well, what are the things that I need to make sure are like T's are crossed and the I's are dotted and that kind of thing.
Yeah, it's so easy to criticize these agencies, but it's a tough
Oh yeah,
thing.
it's new
How
to
do you
them.
do these things?
Like, what are you making?
Yeah, of course. Of course. Well, these things, it's easy. You sit here and we reflect on it and like, oh, wow, look at all what we've done. And it all seems so smooth, right? Like, oh, we had this great idea and we made it and now it's in people and that's great. But I'm sure the path was anything but smooth.
Oh my God.
What was look, there's young people out here, young can be defined however you want, who have their own aspirations to, do this, right? Any words of wisdom or advice or stories of like bumps in the road, and how did you navigate those?
Well, if it's a first-in-class product, like this was, right? Even though it was an old drug, this is like first-in-class. So they had to, there wasn't a mold that you could fit. If you're working on that, expect 15 years at least. So a big fraction of your professional career will be devoted to just getting that one over the finish line. And but it's incredibly satisfying. Even if it fails, I mean, you're learning so much along the way, and there were plenty of opportunities of failure. But I work with many pharmaceutical companies, and I've worked on projects from the beginning to the end with them. And I can tell you that there's always a small group of people in those companies that don't take no for an answer. And so projects are killed all the time, and they never really go away because there's a group of scientists and physicians that believe that this particular mechanism is something that will affect people's lives.
How about you, Karen? Any lessons of the bumpy road you can share?
Yeah, I think along the same lines, it's like, I kind of feel like I'm an eternal optimist. Like every time something doesn't work, I'm like, oh, but I'll do this and it will work next time. So I get back and I'm like, well, actually it didn't work. But it's like, I'm always like, oh, but I'll just do this and then it'll fix it. So I think, that mindset of, you put it in the rabbit and it comes out, but okay, then let's try something different. Or, you think you're going to get a release profile and you don't, but then you just, go back and try it again.
Well, I have more questions I can keep asking, but I want to make sure people came here. We can give them a chance to ask questions. Please speak up. Yeah. Wait, well, Karen will bring the microphone over.
Thank you very much. Very inspiring talk. And I have just a quick question: is if you had any ideas to put some potentially biosensing into this device, even if it could be a platform for this biosensing, and because there are plenty of metabolites in urine, and this is something that is definitely picking up the biosensing world, like Whoop and things like that. So I don't know if we'll be equipped to do that, or if it's something that you are interested in at all.
I can't think of a use case other than, you're always in competition of just taking a urine sample and testing the urine sample, right? So, so is there a need for continuous or, and I just can't think of the use case, right? But if you can identify a use case that
People with sort of diseases with the kidneys that could benefit from.
possibly, yeah. And then you have to match that with the technology and how do you read it and that sort of thing. But yeah, sounds like an interesting idea.
I mean, the device retention part is solved.
Yep.
So I imagine it's just what is actually delivered on the, and can it be delivered and retrieved and whatnot, but yeah, it's an interesting idea.
One of the questions I haven't gotten is, I usually get, is like, can't you tell this is in your bladder? And, I always tell students, some of whom are here in my class, that when you start a project, you have to think of what it is, what are going to be the biggest hurdles, and knock those off. And our biggest hurdle was that people didn't believe that if we put this in somebody's bladder that they wouldn't be able to feel it. So the first human study, they don't.
Just to be clear, they don't feel it. Okay.
Well,
I was wondering
how do we
too.
prove that? Well, we did a study with an empty device and a sham procedure. So we blinded the patients to these were all healthy volunteers. Can you imagine volunteering for this? But
people do this. Just people do this. They get paid.
they get paid.
Not enough for me to do it. But they get
exactly. And we put it in their bladder for I think it was 2 weeks, and then they come back, and those patients that had the device in, it was removed. Other patients, they just did a cystoscopy, and every night they recorded, or they actually had a nurse call them and record all their adverse events that day, any pain that they had. And if you looked at the two groups, they were identical.
And did any of them say they felt something?
Well, that's an interesting thing, because that's different than.
No, I'm asking the question.
Yeah, in fact,
I get there's no difference between the two groups, but that's a different question.
Yeah. If you know you have a device in, then you might say it. And I think there's evidence that people will do that. But if you actually do a blinded experiment, there's no difference between what they report as far as their symptoms. So it's an interesting effect. I mean, you've seen that.
I remember the patient that was most vocal about, yes, I feel it, yes, it's uncomfortable, was in the sham procedure. So it's just like,
Yeah. So if you get it in your mind.
Very interesting. Over here.
So what a great story. Simplicity, I guess. No batteries, no electronics, no complication, and it still works. But two related questions. So the drug is released through this little orifice. What happens if that gets jammed? Say in people who get kidney stones, for example, there would be likely precipitation. And the second related question to that is, how do you know that this thing is working? I mean, what do you test for? Do you just test the urine regularly to see whether it's releasing, or is there something else?
You want to say?
Yeah, so that's a great question with the blocked orifice. That's something that we did a lot of testing on to see, each tube is 100% inspected to confirm presence of an orifice. And then we made some systems purposely with no orifice to see what would happen. And it blows up like a balloon. It just gets bigger and bigger. But we've not found, there is sometimes material on the outside of the system, but we've not found that then occludes the orifice, because you've got this osmotic pressure forcing it out. And the nice thing about our preclinical studies is that they're done in pigs, which are more prone to incrustation. So we're testing it preclinically in the worst case environment. And then the second question I forgot already.
How do you test it?
Oh, how do we test it? Yeah. So in the first The nice thing about gemcitabine is it's a prodrug. So once it gets into the cells, it is turned into an active metabolite. So you can test in the urine. So if there is presence of those active metabolites, the gemcitabine had to get into the cells. So that's one way. So we can measure gemcitabine in the urine and the active metabolites. And patients would collect their urine in the clinical trial. Like they'd have their little jugs that they carried around with them, and they'd have their urine refrigerator, and then they ship it back to us. So the clinical trial really asks a lot of the patients, and that's how we get that data. But we also, in the very first patients that we were testing, since they were already scheduled to have their bladder removed anyways, so the system was put in that time between when they decided they needed their bladder removed and was actually moved, we planned to biopsy the tumor after the bladder was removed and look for levels, but the tumors were gone. So it's kind of like we weren't able to look for levels, but that's a good thing, So it's really looking at, in preclinical studies, we can look at tissue distribution, but in clinical studies, we look at urine. Yeah.
Okay, question back there.
It's amazing that it's such an easy procedure and allow single compartment delivery of the drug. Is there benefit, and is it feasible to make it even more local? Because of using imaging, like endoscope, you can see where's the lesion. Can you apply a device directly on the lesion so that you have a more local drug delivery?
Yeah, well, I think I can.
Even more directed therapy.
Yeah, so it's actually rare that it's just a small region. Like some of these are like carcinoma in situ. It's like everywhere on the surface. A large, not everywhere, but a large section of the bladder.
Spreads across the inside.
Right. So it's actually good that we treat the entire surface of the bladder, because it's very likely that, this is such a recurrent disease. If you just excise the tumor, it comes, you get another tumor somewhere else later. Recurrent, very high rates of recurrence. Yeah.
If I remember the slides, so we will localize lesion, that is not a used lesion.
Yeah, there's microscopic disease. It's unfortunately, that's sort of the nature of cancer, I guess.
But we kind of go with that type of idea if you take the device and then put a targeted drug inside it. Because then even though you're delivering the drug, exposing all of the bladder, it would only impact the cancer cells that's targeted to.
Yeah,
So maybe that's a great way to at least close the formal program is, are you suggesting an antibody conjugate or something like that as a delivery piece? So maybe speak to that, and also maybe both of you, we can start with you, Karen, so what's next? Where are we,
So
what are you most excited about next?
Yeah, so we are working with this as a platform, and we are looking for, how can, we want to treat the whole spectrum of disease. So we're looking at how can we do that? And we're looking at our current clinical trials and learning from, like you were saying, collecting the data, learning from those. And I look forward to really showing a platform in the sense that it's not just one indication that it's approved for.
Yeah,
Not a one-hit wonder.
FGFR mutations are very common in part of the strata there, and so they're using a targeted drug now for that. It's not completely through, but they had published an interim analysis, 90% complete response.
Yeah.
So
Wow.
it's pretty amazing.
That's pretty impressive.
Now it's an interim analysis, so
Yeah.
we'll have to see what it does later,
Yeah.
but it's pretty significant.
Wow.
These are not incremental changes at all.
No, this is a big deal. Well, I guess I just want to thank both of you. First of all, congratulations.
Thank you.
This is life-changing for people, having taken care of patients like this. This is a really big deal, and maybe on behalf of them, let me thank you.
Sure.
I'm sure you guys feel this, all the great things that, but.
Oh, hey.
So thank you.
Thank you.
And just with that, I'll close. I want to thank everyone for coming. Thank you for spending a little bit of time with us here at the Koch Institute and helping us celebrate the amazing accomplishments of one of our investigators and an alumni. So thank you, guys.
Thank you.